It's very interesting how far the field of precision oncology has actually come over the past 10 to 15 years. Now we look at data, we look at technology, different therapeutic modalities, but also our scientific understanding of disease as well, and how it's helping us drive new innovation for patients.
We are in this world of the precision medicine that we determine what is the best for every single patient. We got the tumor, we analyzed the tumor, and I said the tumor expressed particular biomarker, and this biomarker may lead to the different outcome. And according to that, it's kind of bespoke therapy.
Anna touched on something technology-wise, Joyce, and again, maybe you can share a little bit of thought around it. Tell us a little bit about what ADCs are and what's new in this area.
When we look at an antibody-drug conjugate, I think of them in three parts. The first part is the portion of the antibody itself, and that is what actually goes and finds the tumor. And then I'm going to actually go to the end of the antibody-drug conjugate. There's what we call as a payload, the thing that destroys the cancer cell specifically. And then there's something in between that connects them together. So what happens is that ADC goes and finds the specific biomarker or the target on a cancer cell in a specific patient. And while it's going to find it, it's bringing along the payload. Then it attaches to that cancer cell, goes inside of the cancer cell, and then simply put, it blows it up.
So I think what I'm hearing you also say is it a way of trying to basically deliver more precisely what in the past we've seen as being just chemotherapy?
That's right. What we're able to do now is using the biomarker specifically go after the tumor and deliver the chemotherapy only to that cancer cell.
We should generate all the information in the patient's tumor to really tailor the therapy based on that. I think this is the future. I think, one key point is that every single patient should have a molecular testing with all the biomarkers. Because when you have this in front of you, when the patient is newly diagnosed, you learn a lot how you will sequence your therapies. So I think it's equally important, to have the whole picture of the patient when you start therapy.
These modalities help the patients to have another option. And what we're trying to do is not generalize therapy as we've done in the past.
Andrea, there are now different types of technologies that are coming to at least fruition within oncology and clinical research as well. So we hear about different therapeutic modalities, so antibody-drug conjugates. We hear about new types of immunotherapies called T-cell engagers. We also have small molecules that are more directed that we call targeted small molecules as well too. Tell us a little bit about how, again, that is helping evolve how we think about the future of oncology research and how you're applying that, within, your current research endeavors as well.
Yeah, absolutely. I mean, we're getting smarter with the use of these, not only in the development, but also with the use of these technologies and different approaches basically of killing the cancer cells. Historically, the biggest problem with cancer is that it's smart and it gets smarter as we treat it. And so it's very important to have these different mechanisms of being able to effectively eliminate it and kill the cells. We're not just using our typical cytotoxic or sort of typical systemic therapies, but various different ways of targeting the cancer cell through a lot of these technologies, which is just fantastic.
I also want to just say a few words to patients. We're all committed to continuing to help improve outcomes using novel science, technology, data, collaboration to help, again, make a meaningful impact for cancer patients worldwide. Thank you.